I'm going to be talking about some work that my lab has been doing in developing this new tool for finding associations with specific genes with host or community level phenotypes. So I probably don't have to convince this crowd of the kind of general importance of the microbiome. I'll just say really quickly that, of course, we're colonized by very diverse communities at many different body sites. And these communities can influence our health. They can influence human health in a variety of ways. And in some cases, we know this because we can actually transfer the microbes and we can see a change in the phenotype, right? In other cases, maybe these are more associative links. And what I would say is that as impressive as this is and as powerful as this is, we often don't really understand the mechanisms that are driving these associations. And so what we would really like to do is to move from a kind of like taxonomic view to a functional view. So let me start by giving you an example. This is a very typical result that you might get from a sort of case control microbiome study. You sequence a bunch of cases and a bunch of healthy controls. And you look for the taxa that are enriched in one condition versus another, right? So that's great, but it doesn't give you a lot of information on how to interpret this result, right? So one option is that you can simply look at which genes these taxa possess. And so here I'm just showing two genes, gene 1 and gene 2, and you can see that they both look like reasonably okay explanations for this pattern that we're seeing in taxonomic variation. But as you might guess, there is a problem with this, and that's that species are not independent of one another, really. Like, they're related by common descent. And so more closely related species will tend to share both gene content and more similar phenotypes. And so how do we get around this? Well, the solution that I came up with was to essentially wholesale steal a set of methods that were developed in ecology. They're developed in the kind of like late 80s, early 90s by people like Felsenstein and Graffen. These are called phylogenetic comparative methods. And so I didn't invent these, but they are very, I think, underutilized in microbial ecology especially. And the way that these work is that they build this assumption that closely related genes will have more similar phenotypes. In this case, the phenotype is enrichment in cases versus controls. They build that into the model, right? And so what you can see here is that now gene 2, once we know the tree, looks like perhaps a better explanation, or at least an explanation with more evidence, because that gene isn't just limited to one clade, right? We're seeing an association that takes place over more of the tree. And so previously, I put out this tool, Phylogenize, which is designed so that the user can just provide species counts that can be either 16S or from shotgun data, as well as their metadata, and we do everything else. So we give you the pangenomes for all of these species, we give you the trees, and we take care of the statistics and the plotting. That was the kind of version 1 of this tool. And in this second version, there were a couple of issues that we wanted to remedy. And so one of these is that we were using a fairly, at that point, it's a fairly old database of species that mostly focused on isolate genomes. And as we've heard earlier in this session, right, isolate genomes are not necessarily available for most of the environments that we're really interested in. And so in this version, and this is work that's being really driven by Kat Kananen, who's a bioinformatician in my lab, as well as now Nia Tran, who is a graduate student, we've been integrating two very large modern databases that include a lot of MAG genomes. And so these are UHGG, which Chris mentioned earlier. That's a human gut microbiome focused database. As well as a fairly recent revision of GTDB. And so one of the things that we've been doing essentially is annotating these, because we need to know which gene families are shared across species, not just which genes are in a particular genome. And so we've just kind of wrapped up the annotation of these for GTDB. And actually, we're also annotating these with some other functional databases, including KEGG orthologs. And Kat is giving a poster about KEGG annotation right after this session. So another issue in the first version of the tool was statistical. And so as some of you may already be familiar with, one of the challenges in working with metagenomic data is that you mostly have access to relative abundance. So you lose the absolute abundance of different species in your community. And so this leads to effects that are kind of known as like compositionality effects, right? And that makes it difficult to do robust statistics when we're comparing two different environments. And then the other issue, of course, is noise, right? We don't want to get misled by cases where, for example, we saw something once in controls and twice in cases, and then think that this is like a 100% increase, right? It probably isn't, right? And so our solution to this before was we kind of punted and said, okay, we're not going to think about abundance where we have these compositionality effects. We're mostly going to focus on prevalence, which is based on presence absence. And so we developed a method that uses shrinkage. So it shrinks these estimates that are more noisy back to 0 before we put them in the model. And so, but in version 2, we can take advantage of some new methods for analyzing differential abundance of microbes across environments. And the critical thing is that these give us not just the kind of log ratio change, but they also give us a standard error around that change. And that allows us to use a very general purpose method called adaptive shrinkage, where we can, again, shrink these noisy estimates before we plug them into our model. Okay, so in the last few minutes, I just want to give you a little taste of how we've been using this tool. And so this is an application of Phylogenize2 to a data set that is looking at the gut microbiome and liver cirrhosis. And Stephanie, who's pictured here, she's also going to be giving a poster in the next session about a within species analysis of this data set. So this is a more across species analysis. And so previously, there's been this observation that a particular class of commensal Clostridia, which is very prevalent and very abundant in human guts, called the Lachnyspraceae, they tend to be depleted in liver disease. And actually, there's some indications that even restoring these Lachnyspraceae can improve liver function in certain disease models. And so we started to look at this as a reanalysis of some data from Chin et al. And what we found is that indeed, most of these Lachnyspraceae are depleted, but not all of them. And so this seemed like a good use case for us because we can apply this model to try to find genes that explain this difference, right? These differences that we're seeing. And so here's an example of the type of hits that we're able to recover using Phylogenize, as well as with a kind of uncorrected linear model for comparison. So on the left, you can see what's being plotted here is on the left side of each panel is the same phenotype that I just showed you. It's just instead of circular, it's now vertical. So that's enrichment in cirrhosis, right? And then on the right-hand side, this is gene presence absence for a particular significant hit. So on the left side, this is one of our top hits with Phylogenize. And what you can see is that this gene is kind of widely distributed across the entire clade, right? It's not just kind of focused in on one subset of these species. And you can see there are cases where both the phenotype of enrichment in cirrhosis and the gene presence absence phenotype change together in the tree, right? By comparison, the hits that we get with a linear model, they tend to have much weaker evidence. So for example, this is basically mostly a clade marker for this one clade that is like pretty, that it's all red here, that's all enriched in cirrhosis, right? And the linear model does find lots of significant hits, but they tend to be of this type on the right. So these kind of clade markery results. We also compared to a recently published method called POMS, which also does this. And in our hands, we found that in this particular setting, it's not as sensitive as Phylogenize. So it tends to, it may be something about the size of the trees that we're using. And to just tell you about one of these hits super quickly, this was really exciting. We started looking at these results, and we noticed that it included an aerobic ribonucleotide reductase. And so these organisms are really strict anaerobes. I also have a wet lab, and this is like a constant source of frustration is if you have a leak in your anaerobic chamber, they all die as soon as it gets above 50 parts per million oxygen. And so we're really confused about why they have an aerobic version of this kind of essential metabolic gene. It turns out that previous experiments have shown that in a totally different phylum, this aerobic gene actually allows them to have greater tolerance to transient oxygen exposure. And that's something that can happen. It's thought to happen during inflammation. You may have reduced barrier function and also the release of reactive oxygen species from immune reactions that are taking place. And this is also consistent with some other hits that we also see using this method. In summary, we have this tool that we've been working on that uses now these sort of more complete databases and more powerful statistical methods in order to associate genes with a particular environment. We think we have a nice example of the anaerobic oxidative stress response being linked to cirrhosis using this tool. And we're still putting the finishing touches on it, but if you want to use this on your own data, you can either email me or we have a little mailing list that you can sign up for. And I'll just leave you with the acknowledgement slide here. Thank you.
Thank you.